Hello, this is Amanda Welch welcoming you to this Bite-Sized Bio webinar, which today is sponsored by QIAGEN. QIAGEN is the leading global provider of sample-to-insight solutions to transform biological materials into valuable molecular insights. QIAGEN's sample technologies isolate and process DNA, RNA, and proteins from blood, tissue, and other materials. Assay technologies make these biomolecules visible and ready for analysis. Bioinformatics software and knowledge bases interpret data to report relevant, actionable insights. Automation solutions tie these together in seamless and cost-effective molecular testing workflows. QIAGEN provides these workflows to more than 500,000 customers around the world in molecular diagnostics, human healthcare, applied testing, forensics, veterinary testing and food safety, pharma, pharmaceutical and biotechnology companies, and academia, life sciences research. Today's presentation is titled, Using CRISPR-Cas9 to Detect DNA Sequences on Single DNA Molecules with High-Speed AFM, and is being presented by Dr. Jason Reed from Virginia Commonwealth University. Jason is an associate professor in the Department of Physics at Virginia Commonwealth University and a member of the Cancer Molecular Genetics program at VCU Massey Cancer Center. His research group develops nanotechnologies for analysis of living systems with applications in drug development and molecular diagnostics. Prior to joining VCU, he was a faculty member at California Nanosystems Institute at UCLA in Los Angeles, California. He received his PhD from New York University and his BA from Harvard College. Now, as always, we will have a question and answer session after the presentation. So please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Jason at the end. The recording of the webinar will be available at bit.ly/high-speed-AFM. That's bit.ly CRISPR high-speed AFM. CRISPR is uppercase and AFM is uppercase. So now over to you, Jason, for the presentation.
Thank you very much, Amanda. I would like to spend some time this morning discussing a little bit of the motivation for this work, how single molecules can be analyzed with AFM, how AFM works, and then how we're using the CRISPR-Cas9 system to improve the ability to analyze single molecules in systems where you have a small amount of material, which is really the main motivation for this work. So before I begin, I'd like to just quickly thank the folks that are involved. This is a collaboration which has involved a number of different institutions, both at VCU, as well as the University of Bristol and NPL in the United Kingdom, UCLA and NYU. And most of this work was funded by the NIH, either NIGMS or National Cancer Institute. So just to begin to set the stage, most folks in this field are familiar with the Agilent Bioanalyzer, which is a standard benchtop technology for analyzing DNA molecules, sizing them, quantitating them in a relatively rapid fashion, and it's fairly sensitive. It's used across the range of abundances. It's commonly used in preparations, quality control preparations for DNA sequencing and other things, including libraries made from single cells. However, the amount of material is still relatively large compared to the amount of material in a single or a few cells. And as research is going towards wanting to analyze materials from very, very small amounts of input, including single cells and also things like forensic samples, biopsies, whatnot, there is a motivation to come up with a more sensitive technique, and that's where we're applying atomic force microscopy. So what is AFM? AFM stands for atomic force microscope. It's a common method used primarily in material science to analyze surfaces on the nanoscale. So in this slide here shows a typical laboratory atomic force microscope. The device itself, because it's analyzing things that are on the order of nanometers, both in the X, Y, and Z dimensions, tends to be very sensitive to vibrations in the laboratory. So it's usually mounted on a large table, which uses vibration isolation. The actual atomic force microscope device itself is quite small. This red arrow points, in this case, to the head of the AFM, which is a device that holds a microcantilever, which is like a sharp record needle that is very, very small, very difficult to see, probably the width of a human hair, but it's mounted on this fairly substantial platform to control its motion on the nanometer scale in the X, Y, and Z dimensions. At the end of this microcantilever is a sharp tip. The whole thing is usually made out of some sort of silicon material or silicon nitride, which can be manufactured using standard micro fabrication techniques. Key to the technique is the fact that the point of this probe can be very, very sharp. The radius of the tip itself can be on the order of 10 nanometers. And then this tip is then brought into proximity with objects on a smooth surface, and the tip is run over these objects, and as the tip interacts with these objects, it lifts up and down. And then the atomic force microscope uses optical methods to track that motion. And it's actually able to track very, very accurately Z motions on the order of sub-nanometer or angstrom distances. And if it's rastered across the surface slowly, it actually can trace out a topographic map of the objects on a surface. And that's normally what it's used for in material science. It also has a large number of users in the life sciences because it allows nanoscopic scale imaging under many different environmental conditions, liquid, vacuum. It can interact with soft materials as well. So it's a highly flexible imaging system. It also can image single molecules. So one of the things that we have shown is that you can use this method to quantitate DNA molecules or PCR amplicons in a mixture, much like you would do when you wanted to analyze gene expression from several different species at once. Normally, this sort of technique would be done with quantitative PCR, where you have to use separate qPCR reactions for each analyte, which means if you want to do, say, 10 species, and you want to compare that to 10 species in a different sample, then you have to do 20 reactions. Below here, this orange box shows a typical atomic force microscope image of PCR amplicons. The DNA molecules themselves look like little spaghetti strings. They're the light-colored strands. In this particular image, there are actually 10 different species of PCR amplicon, and they have 10 different lengths. The difference, some of the species are highlighted in colors with the names of the genes. And so by preparing a typical mixture of PCR amplicons using multiplex PCR, where there are 10 species in the single reaction, and using a low cycle number where the amplification of the different species is not highly biased based on their size, we found that we could essentially count the number of molecules that would appear in a series of frames and then quantitate their relative abundance. And this was shown to be accurate and also highly consistent with what one could do with multiplex or singleplex quantitative PCR. However, the amount of material required is quite small, and it could be done with only 1 or 2 reactions versus 10 or 20 reactions. Here's an example of some of data from an experiment where a multiplex PCR reaction was analyzed first with a bioanalyzer, which is the data that's presented on the left side of the screen, where 10 species from total human RNA were analyzed, and the relative sizes of the peaks gives the relative abundance of those transcripts in the sample. On the lower left is same analysis of the same transcripts from human brain, and you can see from these two graphs that these genes are expressed at different levels. On the right side is the same sample analyzed using the atomic force microscope technique. However, on the right side, the input material was 1,000 times less. The left side uses the bioanalyzer high sensitivity kit, which is the maximum sensitivity of that system. The AFM easily achieves 1,000 times lower, 1,000 times the sensitivity. You can use 1,000 times lower material. And the data are quite comparable. This shows, on the right side, shows the amplicon links and the different genes. One important thing to note is because the AFM counts single molecules, not fluorescence, the larger molecules appear to be less abundant, but their actual molarity is measured correctly, whereas in the bioanalyzer, because it measures fluorescence, the larger molecules appear to be more abundant because of the total molecular weight is higher. So this shows that it's possible to use this kind of technology to analyze very low abundant samples and do convenient things like simple multiplexing. Problem is that particular analysis takes a long time to gather a significant number of counts from any system because AFM traditionally is slow. A conventional atomic force microscope system that is available commercially typically operates at a data collection rate of less than 50,000 pixels per second. And that is equivalent to taking a typical image in a minute, 2 minutes, 3 minutes. Anyone who's worked with this technique historically knows that that's the major drawback of it. Over the last few years, technologies have matured and a number of different designs for much faster AFM imaging have become available that it would allow frame rates of 2, 4, 10 frames per second over reasonable sizes to be generated. And this is what's called high-speed AFM. So a typical high-speed AFM system would be able to collect on the order of 1 million pixels per second, 2 million pixels per second, which is 50 times faster than a conventional AFM. This makes this sort of molecular analysis of DNA fragments much more user-friendly, and it can be accomplished in a period of time which is usable in a molecular biology lab. So a typical high-speed AFM system, and the one that we use in our laboratory, is a system that is fairly compact, more compact than a normal laboratory AFM because it's much less sensitive to vibration because it operates at much higher rates of scanning. Its data collection happens at a faster rate than the background noise in the environment. So it's much more applicable to be a small desktop box. And this panel on the left side gives you the scale of what one of these instruments looks like compared to a typical coffee cup. The right side video shows how the scanning stage is arranged. In the case of a typical high-speed AFM, in order to achieve these high speeds, on simple samples, the sample itself is moved with what's known as a flexure stage, which allows left and right movements on the order of 1 or 2 kilohertz. That means line rates of 1 or 2 kilohertz, which translates to million pixels per second, and each pixel being 1 to 2 nanometers square. And it still attains the vertical or the height resolution of sub-nanometer. Now, a lot of the advancements are in the electronics to make the noise levels acceptably low in a system with such high bandwidth, and that can't easily be shown here. But that is an important part of the technology. And this is an example of some data. These are live recorded frames. This is the speed at which the system will generate images. And you can see various PCR fragments of different sizes, and some long or unusually shaped objects are actually fragments of genomic DNA that were left over from the PCR reaction. But if anyone's ever used an AFM, you can see that this produces data at a much higher rate. So this higher rate of data production allows one to do things like take very large tiling images. In this case, this large image is about 20 by 20 microns, which is made up of individual tiles of smaller frames, but it can be gathered in just a few minutes. However, the resolution is still retained. So if you were to zoom in, and that shows on the right side shows a zoomed in frame. This frame was captured at a resolution of 2 by 2 nanometers per pixel. So this shows just some genomic DNA from a cell preparation spread on the surface of mica. And one can see that this enables analysis of these kinds of samples, which can be done with conventional AFM, but it's very impractical for a lot of types of uses that one would want to do for molecular biology. Here's another example of a similar kind of image of condensed chromosomes. And anyone that's familiar with chromosome spreads will recognize this image that shows condensed chromosomes on a surface. One of the first applications that we investigated was looking at nucleic acid contamination in spin columns. It was known in the literature a few years ago that through using deep sequencing and some quantitative PCR methods that commercial spin columns from various manufacturers actually contained DNA contamination in them right out of the package. And that's not that surprising because those systems are made to attract and bind DNA. So the question is, how much DNA is really there? How prevalent is that problem? What are these DNA species? So we wanted to look at with AFM to see if we could detect this kind of a contamination. So the simple experiment would be to take a spin column from the manufacturer fresh out of the box, elute ultra-pure water through it, and with that you would be eluting whatever DNA molecules might be contaminating that column. Take that liquid, drop it onto a small mica disc and let the water evaporate, and the molecules will be left over. Image those molecules. In the case of DNA contamination, it's at a very low abundance, so it would require quite a bit of imaging. With the conventional AFM, that would take a very long time, but it's practical with high-speed AFM. These are kind of images you would see. This is what a plasmid digest of known DNA would look like on the upper left. In the middle upper panel, there's a typical small PCR fragment. That's what DNA looks like. Panels C through F show the elutant molecules that are present in several different manufacturer spin columns right out of the box. Now, we have no way of proving initially that this is DNA. However, it looks exactly like DNA in terms of its morphology and its size. So the first thing we did was count the abundance in terms of particle count or molecule count per square micron captured over many images from several different manufacturers, which are labeled here only as A, B, C, and D. And what we found is a few manufacturers had quite high abundances of particles that look like DNA, and some of the manufacturers had very low or close to none compared to control, which was just a water blank. Importantly, most of the abundances in terms of nanograms or picograms per microliter DNA concentration were below even sensitive fluorimeters. So this is not something that would be easily detected using typical laboratory fluorimeters. However, if you were doing deep sequencing from these from samples prepared through these columns, this contamination would show up in your deep sampling. And we were also able to quantitate the size, and the size of most of these fragments is on the order of 100, a little bit less than 100 nanometers, which is 300 base pairs, though some of the manufacturers had quite large fragments on the order of 300 base pairs-ish, which is about 1 kb worth of DNA. For comparison, on the right side, we have some fragments from a plasmid digest to show what the relative sizes would appear of a plasmid digest compared to these fragments. And we went on to do some other biochemistry experiments to show that that material was actually amplifiable using non-specific amplification protocols, which suggested that what we're seeing really is DNA. The problem with these previous analyses with AFM that I've described is the DNA molecules, if they're not any different in length, it's hard to tell what the sequence is, so it limits the utility. One way around this is to actually use the sensitive height detection capability of AFM and label the molecules in some fashion. One simple scheme is what I'm going to call molecular braille, where you are able to bind particles like proteins in some fashion to the backbone in a sequence-specific manner. The AFM then can measure the positions of these particles along the backbone. It's been shown in a number of studies that AFM can measure DNA molecules extremely precisely, almost as precise as a DNA sequencing gel. And therefore, these kinds of patterns that can be recovered from things that you bind to the backbone, assuming that the binding rates are sufficiently high, can create a signature, which then can allow you to trace this pattern back to a sequence database to identify the fragments. And we published some papers on this general scheme in some simple experiments, and here's an example of a simple experiment where streptavidin was bound using streptavidin biotin chemistry on nick sites on a DNA molecule. You can look at the backbone trace generated by the AFM, and you can see the resolution with which these proteins can be localized. On the bottom is a blow-up image of a typical molecule. You can see what this looks like. These markers are easily identified. Part of the limitations of this chemistry, however, is there has to be some way of linking these molecules to the DNA backbone with a high enough fidelity, and we showed in a mathematical analysis that the labeling rate needs to be above 70%, and then the false labeling rate, that is the incorporation of false or false binding, needs to be very low, probably below 10%, in order for this kind of a scheme to be useful in many applications. And streptavidin biotin chemistry with nicking doesn't work as well as one would like for a variety of reasons. However, it does allow the analysis of multiple species on the order of 10 to 15 species in a mixture like we would do in a PCR reaction. And this just shows an example of, let's see, this is 6-7 molecules with their backbone traces from a mixture. And the spikes show the position of these markers along the backbone trace. And this shows an example of a backbone trace of a molecule with 2 markers, for instance. And so this is where the CRISPR system comes in. The CRISPR system, as most people know, is a system which combines a nuclease, Cas9 or some other similar protein, with a targeting guide RNA to create a composite particle, which is part RNA, part protein, but uses a nominally 20 base pair recognition sequence, and that can be programmed by changing the RNA itself and then conjugating it with the protein and then all mixing that with the DNA. And the question we had was that can that be used as a particle system to label DNAs? The advantage being that the 20 base pair being completely programmable allows much more flexibility in the types of loci that one could investigate, the creation of these patterns. However, it was unknown whether one could bind CRISPR-Cas9 to DNA in a stable enough fashion to be able to prepare it and image it with AFM, and also retain the properties of high fidelity, that is, high labeling rates and low false labeling rates. So we did an experiment which proceeded like this, where we have Cas9 with a RNA or sgRNAs that are targeted towards specific sites on test molecules. Those are mixed in a single tube, and then the unbound is removed with a spin column. Those molecules that are labeled are then dropped onto a piece of mica. That surface is imaged with high-speed AFM. This particular cartoon actually shows backbone traces from real images of some long molecules where these red bumps are the Cas9 proteins. And again, you can recover the position of these Cas9 proteins along the backbone and create something like a digital signature that can be matched to a database of DNA sequences to allow us to identify or differentiate molecules in a mixture. Here are some examples of test molecules and then measurements of those test molecules with AFM. So the blue rectangles are actual small AFM images. The red dots on the green molecules are the Cas9 proteins. So we found, surprisingly, that by altering the reaction conditions, even wild-type Cas9 can be made to bind quite stably to DNA molecules such that it survives the purification and imaging process of high-speed AFM. Now, high-speed AFM, because it's moving faster, tends to impart more forces on the surface and would disassociate potentially protein DNA complexes like this. However, we found this Cas9 system to be quite stable. It had very high labeling rates. In most cases, it could be conducted under conditions that would allow labeling rates of 85 to 90% and very low false labeling rates. We conducted a number of experiments with different constructs to measure this, and also to measure the relative sizing precision, which we found to be quite high. Here's an example of not a small construct, but a relatively large molecule. This is a 13 kb long PCR amplicon from one of the exon regions of the gene BRCA1, and it shows these Cas9 proteins bound to ALU1 repeats, which is a general way of using one labeling species to barcode any DNA molecule, since ALU1s and other repeats are very common in the human genome. And the actual known map of this particular sequence is given below, and we can clearly identify that the Cas9 proteins map to the expected sites, and the spacing in between them corresponds to the expected distance. And this can also be done. BRCA1 itself is the locus of it, of the entire gene, is on the order of 100 kb, and so it's quite easy to actually do the same kind of a analysis on a molecule that's 100 kb, in this example of a back clone from the entire BRCA1 locus, labeled with CRISPR-Cas9 and imaged with high-speed AFM. The red bumps along the molecule backbone are the Cas9 proteins targeted to the ALU1 sites, and then the bumps that are off to the side are unbound Cas9s. Because of the precision of the AFM, the bound versus unbound Cas9 proteins are easily distinguished, and the backbone contour length is quite easily measured. This particular 100 kb molecule could be measured in about 2 or 3 seconds at the imaging rates of the high-speed AFM. So it's quite possible to analyze large molecules as well as PCR fragments, which makes this an interesting technique for potentially mapping or detecting breakpoints, structural rearrangements, or other things. The precision of the AFM allows the spacing between different sites to be measured on the order of 10 to 20 base pairs, which means the accuracy of the barcode is quite high, which means in complex mixtures, it's still possible to map these patterns back to the sequence database, which distinguishes this from some other mapping technologies like optical mapping and things like that, where you have worse resolution because you're limited to optical resolutions, and therefore mathematically it becomes difficult when you have a lot of sequences that you're trying to map together. Also, being able to resolve very closely spaced markers allows you to determine features on small scales. So here's this XY graph shows the expected positions of markers on various different test constructs compared to their measured position. It's quite linear. The CVs of the smaller fragments or the smaller spacings on their order of 12 base pairs, and it increases roughly linearly, so the error is about 10% of the fragment size. We're also able to show, because we can measure many molecules with this high-speed system, by averaging, we actually could resolve very small changes. This ladder shows a series of constructs which were 2 base pairs in difference in size, from 140 base pairs up to 150 base pairs. By analyzing and averaging the measurements of many molecules, we actually could distinguish the difference, which shows that this has near single base pair resolution if used in an averaging mode. And this also can be used for things like mapping breakpoints and translocations. So here's an example where we took lymph node shavings from 7 follicular lymphoma patients known to typically have translocations between the IGH locus and the BCL2 locus. We labeled the IGH locus at the J regions with Cas9 proteins, and we labeled the most likely hotspots in a BCL2 with Cas9 proteins, and then we used long PCR to amplify the potential breakpoint areas and map the location. The different patients were found to have breakpoints at different positions within the IGH locus, and that could be determined by the pattern of the molecules recovered from the samples versus what would be expected from the wild type. And we could see that the breakpoints of most patients occurred near the J6 region of IgH. One patient had the breakpoint around the J5 region, and then one point had the breakpoint around J4. In some cases, this breakpoint actually can be prognostic of patient outcomes, and so one could imagine that this could be used in a number of different fashions. So finally, one of the last things we did was show that the technology for high-speed AFM is actually fairly straightforward. It can be implemented even using commercial electronic components, which are quite inexpensive, which means that the system, instead of being an exotic system which would be only found in some core lab, could actually be fabricated and applied to these kinds of analyses in an embodiment that could be on the order of the cost of a PCR machine, for instance. So the typical components of a system like this would use something like a DVD optical pickup. So this optical element is what's in every Blu-ray or DVD player. It actually contains almost all of the electronics and optics required to actually create a high-speed AFM. It contains a lens which focuses a laser beam onto the cantilever. That's normally used to focus a laser beam onto the DVD and record the bits that are coming off the reflective surface. However, it also can be used to record the position of the AFM cantilever, and it has nanometer accuracy. It also has a lot of high-speed electronics because the natural data rates of these devices is tens of megahertz, which is well in excess of what's needed for high-speed AFM imaging. So by simply coupling something like this optical pickup unit with a high-speed flexure stage, one can have a basic high-speed AFM system. And we were able to show in this configuration, we could actually image and easily detect Cas9 molecules bound to DNA. The noise floor of these systems is a little bit higher than a specific, more highly engineered optical system that we were using before. However, the cost of the DVD pickup is on the order of less than $10. So it means that the whole cost of the system is in fact quite low. One can still measure the spacings. This is an example of a backbone of a PCR amplicon from the TERT locus from the human genome, showing two bound Cas9 molecules taken with the optical pickup unit. We did this for a series of samples with TERT and HER2 that had different spacings of Cas9 molecules that we targeted to these PCR amplicons, and showed that we could recover data which looked very, very similar to the more expensive embodiments of high-speed AFM. And so this is one potential way that this technology could be used more widely in laboratories, because it can be made to be relatively inexpensive and easy to use as a desktop instrument. So I will wind it up there. Again, I'd like to thank the collaborators, both students, other faculty here at VCU, as well as our collaborators at the University of Bristol and NPL in London, UCLA, and NYU, who have been part of the development of this technology over years, as well as the funding from the National Institutes of Health. So I will then turn it back over to you, Amanda.
Thanks, Jason. That was a great presentation. We do have a question or so from the audience. If anyone else has a question, please feel free to post it in the questions box that appears on the right of your screen. So the question that we have is about using this with, or comparing this with ChIP-seq. So would it be possible to compare or use that as an augmented method for
ChIP-seq? Yes, that's actually a great idea. One of the ideas behind this is being able to tell what you're looking at. I mean, we clearly can detect low abundance of molecules. The question is, if you pull down some molecules with ChIP-seq, you want to know what locus you pulled down. And so we've shown using in that one of the papers that was cited in this presentation, using a mathematical analysis, that you should be able to identify most loci in the human genome using a generic labeling scheme like ALU1s or some other repeats. which would allow you to identify molecules that were isolated using ChIP-seq, as long as they were several kb long,
Right.
which most gentle prep should be able to isolate. So yeah, we haven't done that kind of thing, but I do think it's quite possible.
Okay, and then we had a question about the resolution for there. So you said something about the resolution being in, as long as you showed the ladder that had, sorry, I'm reading and trying to interpret it at the same time. Um, you said that you had the ladder that showed the difference with two base pairs, and you said
Right.
that
So
you could do that when you were in averaging mode. Could you explain that a little bit more?
sure. So if you take a single DNA molecule with these markers,
Hmm.
um, typically the measurement accuracy, that is the standard deviation of the measurement of the spacing, is on the order of a little bit less than 10% of the distance between the markers. So for
Okay.
100 base pair spacing, that would be 12 base pairs. If you average, with those sorts of statistics, you can see that if you average 10, 20, 30 molecules, you can start to um, improve the precision of the sizing, kind of arbitrarily. We found that actually in practice, one could get down to close to single base pair resolution if you average enough fragments. Typically in a
Okay.
mixture, you would not be averaging, uh, but that kind of thing is possible, and one can imagine you can use that property to look at, certain biophysical changes in molecules and things like that, and resolve those if you're interested in very small length changes and whatnot.
Excellent. So that looks like that brings us to the end of the webinar. So thank you again, Jason, for a very illuminating presentation and a great answer to some of these questions, or to these questions. And thanks also to our sponsor, QIAGEN. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you've enjoyed the webinar and would like to view the video recording of the session, please visit the webinars page on bitesizebio.com. It should be available within the next 24 hours. And there you can see the other webinars we have lined up for you in our free skills webinars. So until next time, good luck in your research, and goodbye from all of us at QIAGEN and Bitesize Bio.